Good evening, everyone. Thank you for joining us tonight for our RiverWatch Fall Speaker Series. For those who may not be familiar with Illinois RiverWatch, we are a community science organization that trains volunteers to monitor water quality in their own local rivers and streams. The mission of RiverWatch is to safeguard the future of Illinois waterways through stewardship, education, and sound science. Now I'm pleased to welcome our presenter, Dr. Jennifer Koop. Jennifer is an associate professor in the Department of Biological Sciences at Northern Illinois University. Her lab studies the ecology and evolution of invasive snails and the parasites that they carry. She seeks to better understand the reasons for their successful invasion, to identify the impact they're having on the ecosystem, and to explore potential management options for the future. So tonight, Jennifer will be talking with us about invasive parasites and the threats that they pose to Illinois waterfowl. And with that, let's welcome Jennifer.
All right, well, thank you so much. I really appreciate the invitation, and I'm excited to talk to you all. I actually really enjoyed learning more about Illinois RiverWatch just in preparation for the talk as well. I'm happy to have anybody interrupt me at any time if you have questions, or if I say something that's just blatantly wrong, feel free to call me out on that too. But I am really excited to talk to you about the work that our lab does at Northern Illinois University. And I've got myself on the title slide here along with my 3 graduate students because I'm going to be talking in large part about the work that they're doing. And all 3 of them are well, maybe 2 of the 3 of them are in attendance at least. So that I'm hoping when we get to the Q&A, I can actually toss the questions to them to be able to answer a bit more about the work that they do. So to just get us started, the way that I usually start these talks, I won't actually make anyone answer this question, but I tend to ask an audience who here has ever had a parasite, and there are usually a few brave folks who will raise their hand and admit that they had, head lice as a kid or something like that. But if you stop and think about it for just a little bit, what you start to realize is that, yeah, you've probably had not just one parasite, but many, many parasites over the course of your lifetime. So things like, hopefully you haven't had fleas, but ticks or mites, or perhaps some gastrointestinal parasites, or things like mosquitoes, all of those are parasites. And part of the reason that you've had so many parasites is that it's such a successful life history strategy. So parasitism is actually one of the most successful life history strategies that we know of. It it is so successful that what it basically means is that every host has at least one species of parasite. Usually they have more than that, which makes for an incredibly diverse array of species of parasites. We think that they may make up over half, or at least half, of all species on the planet. That's a highly contentious number, but the point of this is there's a whole lot of them. These are some of my favorites that I put all on one slide for you so that you can be grossed out all at once. Hopefully nobody's eating dinner, and then we will move past the grossness, I think. So for anyone who's not real familiar with parasites, what I have on here, I'm hoping you can see my cursor. I have things like bed bugs and hookworms and tapeworms, whoops, and other things that I just got rid of. Oh, uh-oh. And now I can't even move my slides. There we go. All right, well, hopefully this stays here. Giardia, I have Giardia down here twice because I like it so much, apparently. Mosquitoes, we have Plasmodium, filarias, lice, lice ferrecing on parasitic hippobossid flies, so meaning that they're moving around on these other parasites between potential hosts. We have fleas and mites. These are some crazy horsehair worms that actually manipulate the behavior of their host, such that crickets end up jumping into puddles of water, essentially committing suicide. They drown, the worms crawl out of their abdomens and sexually reproduce with other worms in that same puddle. You have these crazy cool isopod, copepod type parasites that take over the tongues of fish. So they are consumed by a fish kind of as itty bitty larvae. They grab onto the tongue of that fish and they hang on so tight that they cause it to atrophy. They then stay in the mouth of that fish, feeding parasitically off of any of the meals that it takes for the remainder of its life. So these are just some examples of how diverse the strategy is, the species are, and I'm not even showing any bacteria or viruses or vertebrate parasites. So typically, parasites get a lot of hate, and it's fairly warranted, right? The whole definition of parasitism, excuse me, is that they have a negative effect on their host, usually a negative fitness effect on their host. But it turns out that parasites actually have a good number of positive effects on their ecosystems as well, and they end up being very vital ecosystem components. So here are some positive things that parasites do for ecosystems. At the individual host level, they can actually help to stimulate an immune system of an individual such that they become less susceptible to more virulent parasites. So by getting one parasite and building up an immune response to that one parasite, you prevent yourself from getting another one that might actually be more deadly to you than the first one. At a population level, and this is a really important one for the remainder of the talk, they can help to regulate population sizes of hosts. We are realizing more and more that they are important components of food webs and that they help to stabilize those food webs. So they have a somewhat significant amount of biomass, but they also just have a lot of links between individual nodes or individual organisms in those food webs such that they help to create stability. And then finally, they can also help to increase biodiversity, which brings with it all of the benefits of increased biodiversity in an ecosystem, right? A healthy, functioning, highly biodiverse ecosystem has good ecosystem services, et cetera, et cetera. But sometimes that the heat or the shade that parasites get is actually earned. So sometimes that bad reputation they actually deserve. And it's typically when parasites are also invasive. Um, and the idea of invasive parasites is probably something that you've heard of, but you've likely heard about it as emerging infectious diseases. And while that is a really important realm of research, in general, it focuses almost exclusively on human diseases, so human parasites, human pathogens causing human-related diseases. So I'm going to speak more generally about invasive parasites, meaning any parasite that has moved into a place where it's not considered native, so into a non-native host or into a non-native habitat or region or area. And one of the reasons that it ends up being that parasites end up getting that well-earned bad reputation in that case is because they typically lack an evolutionary history with the hosts that they are affecting in that invaded range. So in their native range, when a parasite evolves some means of attacking its host then potentially co-evolves or reciprocally evolves some means of escaping that parasite. The parasite then evolves some new means of attacking that host, et cetera, et cetera, et cetera, et cetera. But when they lack that evolutionary history, the parasite essentially is given a significant advantage over their host, and they can amplify, that can end up amplifying the negative effects they have on those hosts. Invasive species also find themselves essentially with fewer restraints or with a leg up, more or less, meaning that there's less competition in their invaded range. They might have fewer parasites themselves in an invaded range. All of that can lead to faster population increases or faster range expansion than what you might normally see in their native range. So invasive species are in fact such a big problem that they were named one of the four horsemen of the ecological apocalypse. So this is a term that Jared Diamond of Guns, Germs, and Steel coined. And I really like it because I think it's really effective. It really sticks in terms of getting folks to remember that there are sort of these four main drivers of biodiversity loss in the world today. And I guess to just show cool pictures like this. So one of those is habitat destruction. One is overexploitation, climate change, and then because I found this last guy to be the scariest, I named that one invasive species. So this is not a list that you want to be on. It's a rather infamous list. And again, what it's doing is it's highlighting these four major drivers of biodiversity loss in the world today. We're all familiar with invasive species because we see them every day in almost every single habitat that we either are working or playing in. These are some examples from Illinois. I've got invasive Phragmites. Yes, we do have a native species as well, but this is an invasive Phragmites. We've got the infamous zebra mussel on a boat propeller here, the invasive carp, and then one of our more recent invasive species of concern is actually the spotted lanternfly. And so the reason I've chosen these four to sort of highlight as Illinois invasive species is because they're incredibly prolific. They have moved in and essentially taken over habitats. That is one way in which they can decrease biodiversity. They just sort of outcompete everything else. But what's a little bit crazy is that despite parasites being super common and invasive species being super common, studies of invasive parasites are not that common. And again, I think part of the reason is that where this really hit the ground running was in the world of emerging infectious diseases, which has more of that human component. And I think it left behind a lot of the wildlife world in terms of wanting to understand uh, invasive parasites more generally. So why do we care about invasive parasites? It's because we don't necessarily know whether or not they play by the same rules as invasive species and as other parasites, right? So for example, if we know that invasive species are successful because when they move into a new space, they can outcompete most of their similarly functioning organisms or species. We don't know whether invasive parasites also are successful because they're outcompeting parasites of a similar ecology, right? Vice versa, you can think about it from the parasite side of things, right? Does the fact that they're invasive change the way in which they're parasitic, right? Is it changing how they're interacting with novel hosts in such a dramatic way that that explains perhaps why they are more or less successful as parasites? So in our lab, we kind of have these three overriding lab research goals, and this is essentially the outline of the talk that I'm going to give you today. We're going to walk through first sort of what effects invasive parasites have on hosts, communities, and ecosystems, then how and why are invasive parasites successful, and then finally, how can we better manage invasive parasites? So I'm going to start with what effects do invasive parasites have on their host communities and ecosystems. And throughout the remainder of the talk, I'm going to be discussing it relative to our local research system, or our relatively local research system, which includes the faucet snail and a couple of invasive trematode species, and waterfowl, including diving ducks like lesser scaup. And so this system, I'm going to sort of give you some of its ecology throughout the talk, so I'm not just going to dump the whole background on you right now. You're going to have to hang tight and sort of, I'll spoon feed it to you as we go. But to just start out, this is a species of snail that was introduced from Europe into North America. And we think that it co-invaded with several of these trematode species. The parasites, the trematode parasites, spend half their life cycle in the snails, and then they finish their life cycle in waterfowl, where they can have some pretty detrimental effects. So I was able to collaborate with an art student at NIU who drew this great life cycle for me to try to explain it with some pictures, because I'm a very visual person. So hopefully this helps out anybody else, because it is a complicated life cycle. Apparently I can't touch my computer at all. So what we have happening here is, if we start kind of right up here, we have an infected duck that is shedding trematode eggs via its feces into the water. Those eggs hatch into Miracidia, which are a free-swimming parasitic form, and they go find their way into a snail, typically the faucet snails. These faucet snails are freshwater snails. They get infected with these Miracidia. The Miracidia develop through a couple other stages and emerge again from the snail as free-swimming cercaria. Those cercaria go find another snail. Sometimes they're real lazy and they just go right back into the first snail. Typically, this is also a faucet snail. When they get into this second intermediate host, they develop into something called a metacercaria, which will be important as we move forward to remember that particular stage. This is the stage where it's sort of, these parasites kind of hunker down and just wait in that snail until a duck comes along and eats an infected snail. When that happens, those metacercaria develop into adult flukes, sexually reproduce, and again, complete this life cycle. Unfortunately, in doing that, they tend to have some pretty dramatic fitness effects on those waterfowl. So they can cause pretty severe hemorrhaging that can actually result in death very quickly for these birds. So some of the birds, if they eat just a few highly infected snails, can end up being getting a lethal dose within just a matter of days. And that was in fact how I found out about this system to begin with. So um, I grew up near La Crosse, Wisconsin, but I was living out in Massachusetts at the time, and my parents started sending me all of these headlines that they were getting saying, hey, there's this invasive thing and it's killing all this stuff and you should check it out. And I obviously was like, yeah, I should. So these are just a few little tiny headlines to highlight what was happening in the upper Mississippi River in like the early 2010s, where they were starting to notice high numbers of waterfowl deaths. And when they sent them to the National Wildlife Center in Madison, Wisconsin for necropsy, they were coming back as having died from trematodiasis. So it wasn't high path avian influenza, it wasn't botulism, it was this trematode. And when they looked further at it and started to identify the trematode, they realized this is in this faucet snail. Both of these things look like they're invasive. This is potentially a big problem. So this is about the time that I also jumped on the bandwagon and started doing research with this system. And what we did is we talked to the La Crosse Office of Fish and Wildlife, and in particular, this guy right here, Calvin Gary, who was very clever in that while he was out doing his regular day job, he was making observations of how many waterfowl he was seeing dead and washed up on shore in pools 7 and 8 of the Mississippi River, which is near La Crosse, Wisconsin. He did this for like 10 years, and he had about 13 different data sets going to keep track of all of these observations. And again, he was just sort of doing this in his free time, which was amazing. But what we did is a graduate student of mine, Rebecca Bechtel, got together with Calvin and that La Crosse office, and they combed through all of these data sets to try to get a better assessment of how many birds were actually dying and which species were dying. And when they did that, when they sort of concatenated all of those data sets, what they found was that there had been at least 39,000 deaths between 2003 and 2013 near La Crosse, so in that small area. That was after they removed any sort of repeated things. And again, these are just based on observations, not standardized transects and things like that. They also found that over 15 species were affected by this trematode. And the die-offs since 2013 have been pretty periodic and unpredictable, but none have been quite as severe as what we saw within that first 10-year range. One of the other things that came out of that assessment, though, was the discovery that there are really two species of waterfowl that are adversely affected or disproportionately affected by these trematodes. What I'm showing here on the x-axis, I have pool 7 in the spring is the S, and pool 7 in the fall, pool 8 in the spring, and pool 8 in the fall. So these are just 4 different locations and then 2 different seasons. And along the y-axis, what we've done is we've taken the proportion of all of the mortalities and split them out by what proportion belongs to which species. And I'm hoping what looks fairly obvious is you get this big glob of orange and this big glob of purple. This orange corresponds to lesser scaup, and the purple corresponds to American coot. So lesser scaup and American coot made up the vast majority of observed mortalities, even though we did see mortalities of over 15 different species as a consequence of these trematodes. While I love coot and they're ugly little babies, hunters care a whole lot more about scaup. And so importantly, scaup have been monitored for the past umpteen decades in terms of their populations very carefully because of this hunter interest. And so our lab sort of chose to also focus on the scaup aspect of these die-offs. So I said that scaup have been dying off, and this is just a quick plot showing from the 2014 waterfowl survey the sort of steady decline that is been seen in lesser scaup populations in North America. The explanation for these declines are endless, essentially, but largely they are probably attributed to habitat loss and decline. And to some degree, they may be attributed to the arrival of this trematode species. So while mass mortality events do happen, and that study out of La Crosse showed, that they can be fairly massive in terms of their numbers, it's not likely that those numbers are actually having a massive effect on the total population decline of SCOP. And so what we wanted to do was to look at, essentially, we have reason to believe that there are sublethal effects that are occurring as a consequence of infection by these trematodes. And so Cheyenne Beach did her master's work out of Western Illinois University on SCOP, trying to dig a little bit deeper into what some of those sublethal effects of trematodes might be. So she did this very nice study with captive lesser SCOP in which she experimentally infected them with a sublethal dose of trematodes. So she sort of force fed them these trematode metacercarias, waited 10 days, then euthanized them and measured a slew of body condition type measurements. And what she found was that, first of all, males in better condition were less susceptible to infection, which is kind of interesting. And individuals with the highest parasite intensity lost the most body mass. So that's a pretty important finding in that it essentially tells us what we might have expected, but now have empirical data for, that the higher the parasite load in an individual duck, the more likely it is that it's losing body mass. That may not kill them outright, but it could have a huge effect on their ability to migrate and reach their reproductive or breeding grounds, which could then have pretty significant impacts on population level effects. So Cheyenne began another study that my current graduate student, Scott Herman, is continuing today. Scott's interested in understanding what effects these trematodes might have on SCOP migration. And so one of the things that he did is that he helped to put 100 internal transmitters into, he put internal transmitters into 100 hen lesser SCOP that were trapped in either the Illinois River Valley or in Pool 19 of the Mississippi River, which is essentially right down in here in Illinois. Those birds were then released and their migration pathways were monitored. So these are GPS trackers that feed back data on a fairly regular basis to us. And the idea was that we were going to track birds that would either pass through the known range for faucet snails or not pass through that range, meaning they're either exposed to the trematodes within those faucet snails or they remain unexposed. So we realize this isn't perfect. I'll talk about the not perfect aspect of that a little bit later. But what we're hoping to do then is compare these birds and see whether or not they might have differences in their max migratory distances or in the amount of time that it takes them to migrate, or in whether they manage to actually breed or reproduce once they reach their breeding grounds. So this is a cool map that Scott made for me that each of those individual colored dots is showing you a SCOP as they are doing their spring migration in 2025. So you can kind of see as we get into early March, it's suddenly go time and they all start flying north. And rapid, there's an explosion of colors that happen. That is because all of those dots you saw right at the beginning, those are birds that we actually ended up tagging in 2024. They went north for their spring migration. They then came south for their fall migration, and we got to track them again in 2025 going north. So this is great because we get not just 1 year of data out of them, but multiple years of data. All of the other colors are showing birds that were then given transmitters in 2025. So again, we tracked these birds. We saw whether or not they went through known faucet snail ranges or not, and then compared things like their max migratory distance. And so these are preliminary analyses. Scott is working on writing up his thesis at the moment and hoping to defend in the spring. But whoops, sorry. What this plot is showing these are dots for each of these birds in the exposed group or the unexposed group. So again, exposed, what that means is the birds flew through known faucet snail territory, where they were then exposed to the faucet snails and trematodes, but not necessarily infected. So that is a gap in the study right there. In comparing their max migratory distance, though, it's pretty interesting because preliminary results seem to suggest that those birds that are exposed to trematodes seem to have a shorter migratory distance that they travel compared to unexposed birds. One thing that this study would really benefit from is a ground truth record of existing snail habitat, which is, I'm hoping, where you all come in to some degree, and I'll talk about that right at the end of the talk. And then also in terms of continued study to try to better understand how likely they are to be infected if they do pass through those exposure zones. And so that's all work that Scott is hoping to continue in his PhD. First, I want to explain, though, just a couple of other studies that we have going on in the lab, sort of related to these same kinds of questions. So we're going to move into a little bit more of the how and why are invasive parasites successful. So when ducks move into snail habitat, we really don't know the actual likelihood that they end up getting infected, and that's because there's lots of variability in snail abundance or snail density and in parasite prevalence. And habitat modeling is sort of the ultimate goal here, to be able to make those predictions, and we'll use parasite transmission models to make those predictions. But we need a lot of information gathering before we can do that. And so in traditional models of transmission, typically what happens is the parasite's longevity is often closely associated with that of its host, which means when the host dies, we typically say it is no longer transmitting its parasite. However, when you see these metacercaria in the snails, and remember the metacercaria are the stage in the snail where they are sort of hunkering down and just waiting for a duck to hopefully come by and eat that particular snail. When you see these metacercaria, you realize that they're basically the cockroaches of the parasite world, right? They are built to take on anything. And so what we really wanted to do was test their ability to survive adverse conditions. In particular, what we're considering is desiccation, but one of the things that we're interested in is challenging this assumption that when the snail host dies, the parasites themselves are no longer viable or able to transmit to the next host. So the reason that we chose desiccation as a adverse condition to test the survivability of these snails and metacercaria is that we often see conditions of desiccation occurring along the shorelines of the invasive snail habitat. So these snails are in high numbers, they can get washed up on shore, where they are then sort of left high and dry for a little while. They might then get washed back out to shore, and we don't really know what effect that has on the ability of the snail to survive or on the ability of those metacercaria to survive. So my graduate student, Elise Jordal, created this nice study that she did in the lab using wild-caught snails that were chosen from an area with known high prevalence of trematodes. So they were likely to be infected with trematodes. She then randomly sorted them. I should say my undergraduate, Josh Nelson, who's also in the talk, helped a lot with this project as well. So they sorted them into two different groups, either a control group in which those snails were left in cups of water, simulating sort of their natural conditions, or they were taken out, put on pieces of paper, and left truly to just dry out for a period of 1, 2, or 3 weeks. After those times, they were rehydrated for a period of 24 hours, which allowed them to more reliably quantify snail survival. And then the snails were actually, if they were alive, they're euthanized. All were crushed, the metacercaria were extracted, and then the metacercaria were looked at under a microscope to see whether or not they had survived that same treatment. And again, this is in sort of the preliminary stages. Elise is also hoping to defend her thesis this spring, but some of the major takeaways that we get from this is that she found that snails can survive 3 weeks of continuous time out of water. It's not a lot of snails that survive this, so most of the snails in that sort of treatment died as a consequence of it, but some of them did survive. And perhaps even more surprising is that the trematodes survived 3 weeks of continuous time out of water, even when their snail host died, which that's an important takeaway. I'm now realizing how distracting this figure is, but it made me laugh, and I was trying to pretend that this was a trematode up here. So you'll have to just bear with me. All right, so what are the takeaways from that then? So what I'm showing in these pictures here before I do this looks a little bit like sand in between these big rocks here, but if you zoom in, what you can see is these are actually all little tiny faucet snail shells. So we can find entire shorelines that are essentially made up of huge numbers of these faucet snail shells. All those snail shells that are washed up on shore might still be able to transmit that trematode to ducks if they consume them, especially if the shells are basically washed back into the water or get stuck in vegetation mats. So for the most part, scaup are not coming to the shore and consuming snails off of the shoreline. They're diving ducks, right? They have a different feeding ecology from that. But if those snail shells are washed back out in vegetation mats and put back into the water column, then it is increasing the potential that those birds accidentally consume those snails and then can potentially be infected. So what that means is that we expect the abundance of trematodes to sort of continuously increase over the course of a season, even if the snail populations remain constant. And that absolutely challenges the way that we traditionally set up parasite transmission models. So that'll have important implications as we move forward to that next step. Okay, to move a little bit more into the why, right? So why are these snails, why are these parasites so successful? This is certainly a little bit more of the theoretical aspect of our work, but I think it's really important, and it actually plays right into why we need the help of folks like Illinois RiverWatch with our system. So this is a plot with these little orange squares showing the prevalence of faucet snails across the globe. Again, they're a freshwater snail, so largely just found on larger land masses. They are native to Europe. They invaded North America in the late 1800s, early 1900s, likely via ballast water. This is an animated map from USGS that is showing all of the official records of faucet snails in North America, largely just in the US. But what you can kind of see is that there's sort of an east to west progression through the Great Lakes. So kind of from the Hudson through the Erie Canal down through the Great Lakes. They hop, skip, and jump over to Montana, where likely via recreational boats. And then in the mid 2000, 2005 to 2010, they all of a sudden pop up in the Mississippi River. And that is where we are currently seeing, and then it just explodes after that because we realized this is a big deal and we went looking for them, and voila, we find them everywhere, right? This is typically how invasive species research goes, is they fly under the radar for a period of time until they start to have a pretty significant effect on a species that we monitor. And then all of a sudden we find that invasive species everywhere. So currently, faucet snails are moving south down the Mississippi River. And one of the things that we wanted to do was to understand a little bit more about their population structure in both the invaded and native habitat. And the reason to do that is because when we ask questions like, why are they so successful in North America, right, in this invaded range, what we often want to do is immediately compare traits between their native range and their invasive range. But if we don't know whether those are actually considered separate or unique populations, those trait comparisons aren't always necessarily fair. And so in doing this first, what we can do is say whether or not hopefully we can tell where in Europe these snails came from. We can also then tell how they invaded in North America, right? Whether it was multiple introductions or whether it was a singular introduction that spread from there. That sets us up for far more informed comparisons of traits or fitness consequences on hosts, et cetera, in our future studies. So this is work that my graduate student Tristan Chavez-Poschel is doing right now. He has collected samples of snails and their trematodes from multiple places across Europe. Sorry, we got rid of the Atlantic Ocean here and just slammed these two continents together. but multiple places in Europe, and then from essentially throughout the invasive range in North America, minus Montana for right now. And so he will be using a series of genetic tools to understand the population structure of the snails and the trematodes such that we can ask those next step questions. A previous master's student of mine did a preliminary analysis already on some of the snail DNA that we had from the Netherlands, from Lake Oneida in New York, and then from each of these different pools in the upper Mississippi River region. And you don't really need to understand what a structure plot is in order to get this. I'll try to explain it, but essentially each vertical line here is a snail. So you get a snail, you extract its DNA, you send it off for essentially whole genome sequencing. You get those sequences back from all of your snails and you put them into a computer program and you say, okay, look at these sequences and tell me which population you think they belong to. And you tell me which one they belong to if there are 2 populations or 3 populations, et cetera, et cetera, et cetera. And then you can compare those models to find the ones with the most support. When we did that, we found the most support for there either being 2 populations or 3 populations out of all of these sampling locations. But the biggest takeaway here is that essentially all of the snails in the Mississippi River look very genetically similar. Those in Lake Oneida out in New York look a little bit different, right? It looks like they maybe share half their genetics with those in the Mississippi and half their genetics with those in the Netherlands. It's a little bit misleading, but it's the same idea. And those snails in the Netherlands look completely different, right? So this lets us know that yes, we have genetic differences between these populations, but not within the Mississippi necessarily. So comparing traits between Mississippi populations may not be fair because they may all be genetically more or less identical. So Tristan is going to continue this work using all of those new sampling locations that he has and some refined techniques that we have as well. He will be doing this both with snails and with their trematodes. And once we create these structure plots and these assessments of where the populations are, we can actually also do some Bayesian stats to try to figure out what the pathway of invasion looked like. So much like a phylogenetic tree shows where a species splits off from another species, these invasion pathway plots show when one invaded population split off from another invaded population, and whether or not there was sort of mixture after the fact or not island hopping, but I forgot the word for it, where one feeds the next, which feeds the next, which feeds the next invasive population. So that's exciting work that he will be doing as he finishes up his PhD, and I hope to report back on that. All right, the last part of my talk is to talk to you all about how we can better manage invasive parasites, and that's really where you all come in. So one of the things that we really need to be able to do to effectively study this system is to track its current invasion. We're incredibly lucky in that the invasion has moved as slowly as it has. So again, they were introduced in the late 1800s, and we still are only seeing them about as far south as pool 13, 14, or 15 of the Mississippi River. So they are moving at a snail's pace throughout their invasive range. That means we can keep tracking them, right? It's not that hard to get ahead of them. So again, we've been tracking them in Illinois waters to some degree, but we are limited in our capacity to do so just because we are one lab and X number of people. We know that faucet snails are in the Illinois rivers, so they're actually in the Mississippi River, again, about as far south as Pool 15, and they are in the, I always say this wrong because I'm from Wisconsin, the Des Plaines River over on the east side of Illinois as well. We found them near the Four Rivers Environmental Center. We haven't found them in a lot of other places in Illinois, to be honest, which is good news. But it's also largely because we haven't had the opportunity to really go look for them. So this is where we would love a little bit of help if folks are out in the rivers and they want to hunt for some snails, they can do so. So kind of the ideal habitat to look for these faucet snails are riprap areas. So those are areas of like man-made shorelines with kind of bouldery rocks. Typically, you want to find riprap that has emergent vegetation near it, so either wild rice or wild celery beds. We also find them in higher nutrient, low flow type areas. So it may be that there's no riprap, there's just like kind of a silty sediment. But if there's that emergent vegetation and a little bit of flow or a little bit of movement to the water, then there's potential of finding these faucet snails in those areas as well. So sometimes you find them on, floating log debris and things like that. Faucet snails are relatively easy to identify because there aren't a huge number of lookalikes. So they are only about 8 to 12 millimeters full grown. So you're looking at like a centimeter in size for most of these snails. And one of their super defining features is that they have an operculum. And the operculum is this piece right here on the snail. It's a little bit like a trap, not a trap door, but a door. So that when they experience adverse conditions like desiccation, they can close themselves up, seal that door, and just wait out whatever is happening on the outside world, right? So if you take one of these snails and you flip it over and you try to look at the snail body, they will close up and pull that door closed and you won't be able to see it. So that's what the operculum is. Their color varies a lot though, from light tan all the way to almost black, depending on how stagnant the water is or what's growing on them, and actually even the type of season. But they always have these 4 to 6 whorls. So this particular picture is from USGS, but it's sort of missing its last whorl. So you got 1, 2, 3, 4. We almost always see a fifth one right up here. It's almost like this one got knocked off just a little bit. So 4 to 6 whorls on them. Some of the lookalikes, so I've put faucet snails over here on the far left as a reference, but some of their closest lookalikes are the Amnicola mud snail, which they also have an operculum. In this one, it's shown far recessed, but often it sits right here, just like it does in the faucet snail. The trick with Amnicola is that they're really tiny. So their maximum size is half of what faucet snails is, so only about 5 millimeters. And they have fewer whorls as well, so typically only 3 whorls on them. If you go out and you find a whole bunch of snails that all look like faucet snails, but they're all about 5 millimeters in size, it's more likely you found a bunch of Amnicola than you found a single juvenile class of faucet snails. The acute bladder snail, or Physa, can have a somewhat similar shape, and we find them in almost the exact same habitat all the time. They're easy to decipher in that the Physa don't have that operculum. So when you flip them over and look at the snail, you can see this little snail just being like, leave me alone, right? They don't have a door to close up. So those are a different species as well. The New Zealand mud snail, unfortunately, is an invasive species that has almost the same invasive range right now as faucet snails. But again, they are very tiny. So their max size is about 5 millimeters. They can reach really high densities, though, just like faucet snails. And I didn't know how to describe this, and I'm sure that there's some molluscologist that's going to be mad at me, but they're pointier. I'm sure there's a term for that in the snail world, but I don't know what it is. So they are pointier. And then finally, we have these our mystery snails. So the Chinese, Japanese, and banded mystery snails, which have an extremely similar shape to the faucet snail. They all have an operculum, but they are all chunkamunks. So they are massive. The Chinese mystery snail grows up to 10, maybe 15 centimeters in size. The Japanese mystery snail is somewhere between 8 and 10, and the banded mystery snail is kept between 4 and 5 centimeters in size. So these are much, much bigger than any of the faucet snails that you'd ever be able to find. So should you discover any of these in your adventures, I would strongly encourage you to report back. So you can send photos of any of these suspicious collections to me at Northern Illinois University. It's really, really helpful if you send photos that include some sort of scale. So throw a ruler in there, or throw a quarter, a nickel, or whatever you got in your pocket in there for a little bit of scale. That helps us in identifying those snails. And then if you can show the operculum slide up, that is also very handy for us. So with that, I have lots of folks to acknowledge in terms of collaborators at the Illinois Natural History Survey. So Dr. Ariel Fournier is the co-advisor with Scott Herman, and she has been tremendously helpful in organizing all of these studies. Lots of other collaborators throughout different agencies, some former lab members whose work I've shared, and then our funding agencies, which include the Illinois DNR, and NSF, and Ducks Unlimited. And so with that, I and my grad students would be happy to take any questions. Thanks so much.
Thank you. That was a great presentation. I see that we already do have a question in the chat. Go ahead and pull that up here. So Patricia is asking, how is the European waterfowl population? Has this been devastating to certain European populations? And is there a way to diminish the spread or invasion/infection for our US populations?
Yeah, that's a great question, Patricia. So, Elise, you just did this, right? You just did your history of waterfowl deaths in Europe. You want to speak to that?
Yeah, I don't mind. So, oops, sorry, but I just bugged my dog. He did not like that. Um, so one thing that is really surprising with the European population is there's actually not really very much literature on it. And so one takeaway from that is that it's not happening that much. So it's not people aren't publishing on it because it's not happening, but um the scientific reason, the really big important reason behind it is that um the birds and the parasites were in the same environment, and so they have kind of gone back and forth and co-evolved together so that the birds aren't as affected by the parasites in Europe. Whereas in North America, it's kind of it's like a foreign body almost, and the birds in North America have no idea how to deal with that. They've never had any previous exposure, and so they're kind of like, what do I do? And they die. Um, whereas the birds in Europe have had this previous exposure, and they maybe have antibodies from their parents, or something like that. But it is it's a pretty big gap, actually. If I was European, I would dig into it.
Yeah, thanks, Elise. Yeah, we do know that the faucet snail in Europe is host to a slew of other trematode species compared to what they have in North America. So in North America, they really only host about 3 different trematode species, and in Europe, I think it's close to 30 different trematode species that they have. So there may be some competition happening within faucet snails, such that we see very different effects when those faucet snails are consumed by waterfowl in Europe. So yeah, short answer, we don't think that they're having any sort of devastating population level effects on European birds. In terms of whether there's a way to diminish the spread or invasion within the US populations, that is a very tricky question. Short answer, no, there really isn't. So these snails are continuing to move south. They're doing it slowly, but they are continuing to move down the Mississippi River. They are jumping into new lakes and ponds almost every day. We think that those are popping up. And unfortunately, the habitat that is ideal for those snails is ideal for our waterfowl. So one of the regions in Pool 7 where we found so many of those waterfowl deaths was actually a waterfowl refuge area. So it means hunters can't go in that area during hunting season. You know what this means. But uh, that is the ideal habitat where we also find super high populations and densities of these snails and the parasites. And so the only way to potentially mitigate the effect would be to exclude birds from that area. And that then just puts those birds at further risk from things like either a lack of a food supply or being exposed to more hunters. So it's a very tricky situation that we don't have a solution to right now.
Okay, and then a follow-up, Patricia was asking if there's any possibility of human infection.
Not that we know of. There's no evidence that these trematode species use mammal hosts in any way. There are plenty of trematodes that do that, that can go both between yes, there's some that can go between avian and mammalian hosts for sure. But there's no evidence that these particular species do that, so there's no reason to think that they would jump into humans.
Um,
Uh, oh,
Go
sorry.
ahead.
Oh, I was just gonna add they're really sensitive to the pH, so they actually can't even infect chickens. which is kind of crazy to think about. It's really very, very sensitive to the pHs in the gastrointestinal tract of the birds.
Yeah. People eating poorly cooked ducks, good question. I would be very surprised. I'd be very surprised if those, one, you'd have to basically eat the intestines of the duck. That's where all of the adult worms are, or really, really poorly clean your duck and then really poorly cook it. Might expose you to some of those trematode parasites. But again, there's absolutely no evidence that they are likely to go into humans. So I don't think that's a risk that we even need to raise. It's a great question, though. Very fair.
Something that I'm kind of wondering is that you mentioned at the beginning that there were, I believe, 15 different species of waterfowl that had been recorded. There had been deaths recorded and associated with these trematodes. But really, most of it was just those two species. So I guess what's going on with the other 13 species? Is it that they might eat snails but not often? Is it that they for some reason have an immunity of some kind?
Yeah, that's a really good and insightful question. Um, yes, to some degree, it's that some of those species are not molluscivorous, so they're not purposefully feeding on snails and other mollusks. So they're sort of accidentally consuming them, right, in vegetation or seeds and other things like that. They happen to get a couple of snails and then get infected. There's also likely the case to be made that there are birds with more similar trematode parasites that affect them such that they may already have immunity towards these. That hasn't been studied. I don't think anybody's really looked at it to see why there is the difference in susceptibility. The other thing that I would caution against is when Calvin was out there doing these assessments, he was doing it at the times when Scop and Koop were actually in really high densities. So it may just be more a reflection of the overall densities of those birds or the abundance of those species of birds rather than their susceptibility to the parasite. But he didn't find, copious numbers of mallards washing up and things like that. So those are another bird that are always in high abundance in those same regions, and he didn't find those. Or teal also are another one that we didn't find high numbers of. So it's a little bit of a mystery. It's a good question, though.
Great. Are there any other questions from the audience? Okay, well, not seeing any other questions, so I guess we can close this out tonight. So thank you for this great presentation, and thanks to those who joined us this evening. Before you go, I do want to mention that our next speaker series event will not be until January. So watch our newsletter or our social media for details. Once again, thank you for joining us tonight and have a great evening.
Thank you so much. I appreciate it.